Good evening and welcome, everyone. It is wonderful to see so many familiar faces here tonight and to meet new ones as well. On behalf of the LAM Foundation, it is my pleasure to welcome you to this year's Day of Giving Advancing LAM Science panel discussion and our celebratory social hour. My name is Cindy Beasley, and I am the Director of Education and Advocacy at the LAM Foundation, and I will be your moderator this evening. We are delighted to have you join us. This evening's program is the final highlight of our 2024 Day of Giving, the most important fundraiser of the year. That's because 100% of funds raised this evening go directly to research. This year alone, we received 27 letters of intent to propose a study for LAM research funding. These letters will be reviewed by our Scientific Advisory Board in late fall and brought to the Board of Directors at their December meeting for 2025 funding. The funding raised today directly supports any grants awarded. Now, while interest in LAM Science remains high, we are limited only by the amount of funding received. Your gifts have a direct impact on advancing LAM Science. Without this funding, there would not be a treatment for LAM today. Now, I don't know if you're aware, but the LAM Foundation grant model is one of funding pilot projects. Scientists approach the LAM Foundation with an idea to advance LAM research. Upon scientific review of the research proposal, to ensure it aligns with the Foundation's mission and research goals, proposals are then ranked. The number of grants awarded is dependent on how much money we raise. This has an amazing multiplier effect. As the research advances, scientists use the data collected from Foundation funding to apply for larger grants. These larger grants result in more research, which leads to more data, which will ultimately lead to the breakthroughs we need. Your dollars lead to more dollars to study LAM. In fact, the LAM Foundation's model of research funding has resulted in a 7-fold increase in LAM research dollars. Now, it's easy to give. Simply go to thelamfoundation.org/donate. The panel discussion tonight will highlight LAM Science advances and the potential they hold for therapies in the future. Investing in hope is not merely a pipe dream for the LAM Foundation. We have a proven track record of turning hope into reality. We know it takes time, we know it takes perseverance, but we know breakthroughs are possible. Our panelists tonight are at the top of their respective fields. We are fortunate to have them share their knowledge and expertise with us. Now, before we begin, we have a few housekeeping items to take care of. Microphones will be muted and the chat box disabled throughout the presentation. At the conclusion of the presentation, we will open up the chat box for Q&A. Now, this evening's program is research focused. The chat box Q&A is to be used for research questions only. Unfortunately, we will not be able to address personal or clinical questions during this session. Questions that were submitted in advance of this evening will be covered first. We will do our best to answer all other questions. Then following the Q&A, we will immediately move into our celebratory social hour. I do hope you will join us with a beverage of your choice as we toast our successes and one another. And now for the good stuff. There is a saying in the research sphere that we stand on the shoulders of giants. This phrase is often used to symbolize scientific progress and the idea that new ideas are built on the work of those who came before us. This evening, we have 3 of those giants in LAM research joining us. Each of them have moved LAM science forward in unexpected ways based on their pioneering work. Many of you know Dr. Krymskaya because of her long association with the LAM Foundation. The best way to describe her is as a trailblazer. In 2002, she discovered the function of the gene which causes LAM. Her pioneering research on LAM paved the way for the FDA approval of Rapalogs as a new treatment for LAM and TS. In 2008, she was the first female scientist appointed as tenured professor of medicine at the University of Pennsylvania School of Medicine, the first medical school in the United States. I also want to point out that excelling as a research scientist involves mastering a highly specialized and demanding field. It requires not only understanding complex scientific concepts, but also contributing original research and ideas. Dr. Krymskaya's ability to achieve this level of success while navigating a second language adds an additional layer of accomplishment and speaks to her brilliance. We are fortunate to have her leadership as scientific director of the LAM Foundation. Dr. Krymskaya?
Good evening. Thank you for a lovely introduction. As a scientific director of the LAM Foundation, I'm honored to address you all tonight. I feel immensely privileged to advocate for and serve LAM community. I want to assure you that the LAM Foundation is committed to our mission of enhancing the lives of LAM patients who are at the forefront of all our endeavors. I also must recognize the incredible legacy of the LAM Foundation, which will be celebrating 30 years in 2025. On behalf of the Board of Directors, I wish to express immense gratitude to our founder, Sue Barnes, whose unwavering dedication and passion paved the way for us to gather here tonight. Additionally, Dr. Frank McCormack, our founding scientific director, deserves our deepest appreciation for his tireless commitment to advancing LAM research and patient care. Tonight, I'm asking and taking this opportunity to ask to donate to the LAM Foundation to sustain our research mission. Your support of LAM basic translational and clinical research is invaluable. Funds raised tonight provide essential seed money to initiate laboratory and clinical studies to test new hypotheses that otherwise would not be pursued. Your contributions enable the generation of critical preliminary data, allowing researchers to apply for larger federal grants. On a personal note, I'd like to share how seed money has impacted my own journey with LAM. Over 2 decades ago, I approached Sue Barnes and Dr. Frank McCormack to express my interest in conducting LAM research. Following Frank's guidance, I applied for and received $25,000 pilot award from the LAM Foundation. This initial funding allowed me to conduct key experiments leading to the discovery of the function of the TSC2 gene, which causes LAM. Based on these findings, I secured a $1 million grant from the National Institutes of Health, which enabled preclinical testing of rapamycin to inhibit the growth of LAM cells. This pivotal data paved the way for rapamycin clinical trials and ultimately FDA approval of rapamycin. I'm excited to announce that the LAM Foundation's annual grant cycle is underway. With your support, we aim to fund research in the following priority area as part of our patient-centered LAM research focus, including understanding the role of hormones in LAM. We urgently need advances in this area to address critical questions, including how do hormones trigger LAM cell growth, leading to lung destruction and their spread to lymphatic and kidney over years in young women without visible symptoms? Why does pregnancy accelerate lung destruction, leading to spontaneous pneumothorax and chylus complications? How do hormones impact ongoing lung function decline in LAM patients on rapamycin, advancing some to the need for lung transplantation? We also need to develop new treatments beyond sirolimus. We're actively seeking alternatives to sirolimus, exploring other options that could be used alone or in combination with rapamycin. In addition, advancing diagnostic and prognostic biomarkers is critical for better assessing disease onset using blood tests, tissue-based assay, AI-driven histology analysis, and tracking and predicting the course of LAM. These advancements will also enhance monitoring treatment responses in individual patients and facilitate new clinical trials. We also understand the importance of conducting discovery research. by utilizing available and novel state-of-the-art technology, including AI, to get deeper understanding of LAM etiology. These approaches will help us to identify novel mechanisms in LAM pathogenesis that could serve as a therapeutic targets beyond mTOR pathway, especially for rapamycin in unresponsive LAM patients. We're also looking into applying multiomics and AI approaches to identify proteomic patterns in LAM patient serum as diagnostic, prognostic, and treatment response biomarkers. Developing cellular in animal model also help us to perform new experiments and test new drugs for treatment of LAM. This year, we have also introduced the Clinical Research Award to support investigations addressing unmet clinical needs relevant to the care and quality of life of LAM patients. In closing, I view the effort of the LAM Foundation, along with dedicated scientists and clinicians, as a collaborative mission fueled by a shared commitment to prolonging lives and improving the quality of life for women affected by LAM. In both clinic and lab, it's easy to become absorbed sometimes in procedural or technical challenges and maybe even lose sight of human aspect of LAM. Therefore, ensuring that the patient's voice remains central to our work, whether in research or clinical care, is absolutely essential. This underscores the critical role of the LAM Foundation in advancing scientific understanding and clinical care to discover new treatments and ultimately pave the way towards a cure. We deeply appreciate your support in these endeavors. Thank you.
Thank you, Dr. Krymskaya, for your leadership and inspiration in advancing LAM science. We are so appreciative of you and look forward to seeing LAM research develop under your guidance. Now it is my pleasure to introduce two more giants in the LAM research sphere. Dr. Lisa Henske is renowned for her discovery that mutations in the TSC2 gene cause the sporadic form of lymphangioliomyomatosis. Her discovery provided the basis for key clinical trials demonstrating the effectiveness of mTOR inhibition with sirolimus rapamycin for the treatment of LAM. Dr. Henske is a physician at Brigham and Women's Hospital, where she is director of the Center for LAM Research and Clinical Care. She is also professor of medicine at Harvard Medical School and a medical oncologist at the Dana-Farber Cancer Institute. She has received grant funding from NHLBI to study the molecular and genetic underpinnings of LAM, as well as explore novel therapeutic strategies for LAM. She has received awards for her research from the LAM Foundation, the Tuberous Sclerosis Alliance, the Society for Women's Health Research, and the American Thoracic Society. Dr. Stephen Hamas is the Lewis S. Wolk Distinguished Professor of Medicine, Chief of the Division of Endocrinology. I want to make sure we see him. Yep. Diabetes and Metabolism. There we go. And Executive Vice Chair of the Department of Medicine at the University of Rochester in Rochester, New York. He is also the immediate past president of the Endocrine Society, an international organization of over 18,000 members. He received his MD and a PhD in immunology and microbiology from Duke University, followed by a residency in general medicine and a fellowship in endocrinology at the University of California in San Francisco. His laboratory has developed a mouse model for LAM and is currently examining the roles of estrogen as well as inflammation in disease progression. So welcome, Dr. Henske and Dr. Hamas.
Thank you so much, Cindy. Are you seeing my slides?
Yes, we are.
Great, wonderful, thank you. I'm very, very happy and honored to be here on this special night. I'm going to tell you just a glimpse of some of the advances in research that I am personally excited about, and the advances I'll be telling you about focus on the immune system. So everyone here already knows what LAM is, but just to put us all on the same page, LAM can cause three different things to the lung. It can cause lung collapse, fluid accumulation around the lung, or damage to alveoli, which are the delicate air sacs that I sometimes liken to the cherry blossoms. This is a photograph of the cherry blossoms in Washington, D.C. And when these cherry blossom-like air sacs are damaged, that's what we refer to as cysts, because they look like holes on a CT scan. So the question that Vera already raised is what treatments could eliminate rather than suppress LAM cells? So this is the amazing clinical trial, a huge, huge breakthrough in LAM that was led by Frank McCormick, who is one of the most amazing people I've ever known. And this trial, which is you'll hear us refer to it as the MILES trial, which stands for Multi-Center International LAM Efficacy of Sirolimus, showed that in women who have LAM, who were randomly assigned to either receive sirolimus or placebo, that the women who received sirolimus with the blue squares here, their lung function, as measured here in the forced expiratory volume in 1 second, FEV1, stabilized during 1 year of treatment, while the women who were receiving placebo had ongoing loss of lung function during 1 year of treatment. And then when the drug was stopped, lung function declined in both groups. Now, if we had a perfect treatment that eliminated the LAM cells altogether, when we stopped the treatment, LAM would not continue to progress. So we would love to find a treatment like that you could take for a year and then maybe have a long treatment break or perhaps never need treatment again. So how can we get there? Well, I'm going to tell you about some ideas that relate to using our body's own immune cells. I'll be talking specifically about 2 types of immune cells called T cells and macrophages, using those immune cells in our own bodies to recognize and eliminate the LAM cells. I'll be telling you about this from the perspective of research that we're doing in my lab, but I want to really highlight that this is not just my lab that's doing this. Actually, Dr. Krymskaya, who we are so incredibly lucky to have as our scientific director, is also doing research and has done amazing research in very similar studies. And so have many other people, including Steve Hamas, our next speaker, Caroline LaPoule, Simon Johnson, Joel Moss, Katerina Meisel, and many investigators at the University of Cincinnati, Jane Yu, Yan Zhu, Frank McCormick, Minzy Go, and others. So it's an area that we need a lot of people. The immune system is very complicated, and we're fortunate to have a lot of people who are looking at this from several different directions. Okay, so I think that we've been thinking about LAM for quite a while from the perspective of the LAM cell. This is a LAM lung biopsy, and these cells right here that are sort of the darker purplish color are the LAM cells. But there are lots of other cells in the lung in someone who has LAM, and I think we're just starting to understand what else is going on. So I think of this as in the box and out of the box. And everything I'll be telling you about today is out of the box. Basically, how is the LAM cell remodeling the cells around it? And does that matter for the disease? So I'm going to start by talking about T cells. T cells they became pretty famous during COVID and even made it into the New York Times crossword puzzle earlier this month. A small component of an immune response, a T cell. So T cells get their name because they are partly derived from the thymus gland, which is in the chest. And they have this amazing ability to bind to tumor cells and other things that shouldn't be in your body. And when they do that, they can release these little packages of toxic compounds. They can direct those toward a neighboring cell that they have recognized doesn't belong there and destroy that other cell. And this is a really important role that T cells have. They're going around the body looking for things that don't belong. So a very wonderful scientist working with me, Tina Liu, has made 2 discoveries about T cells in LAM that I'd like to tell you about. This was the first study published in 2018, where she treated mice that we were using as a model of LAM with drugs, including an antibody to a molecule called PD-1 that we use very commonly in oncology these days, and found that the mice responded to those treatments. And the reason that they responded is that the T cells are exhausted. Now, you might think that exhausted is like a lay term, but it's not. Exhaustion is the term that cancer and tumor immunologists use to designate T cells that are no longer in this active state, but have seen something else so many times that they think, maybe I shouldn't worry about that anymore. So in the case of LAM, we found that the LAM cells are inducing this T cell exhaustion where the T cells are still there, but they are no longer recognizing or destroying the LAM cells. In fact, this occurs very often in cancer, but I think this might have been the first time that T cell exhaustion was discovered in a benign tumor situation like LAM. So we didn't know at the time exactly why the LAM cells were causing this exhaustion for the T cells, but Tina made a second really, what I think is a super interesting discovery. It was published in 2023, where she found that the LAM cells are coated by a protein called B7H3 that makes them invisible essentially to the T cells. And these are the lungs of a mouse that we use as a model for LAM. And she found that if we remove this protein B7H3 from the LAM-like cells, all these little nodules in the mouse lung were gone. So we saw this in another model using cells that were actually developed by Vera Krymskaya. So what we think is happening here is that it's specifically this protein on the surface, B7H3 protein on the surface of the LAM cells, that is causing them to be essentially invisible and exhausting these T cells. And when we remove the B7H3 from the LAM cells, suddenly these T cells are active again and are able to recognize and destroy the LAM cells. So now I want to talk about a second study by another wonderful scientist who I have the privilege of working with, Yan Tang. So you saw this image earlier of the LAM cells surrounded by a lot of other cells, and that makes it pretty hard for us to figure out exactly what's going on. It's kind of like having a big jar of M&Ms all mixed together. And if you really wanted to know how many blue M&Ms there were and what they're doing and where are they relative to the red M&Ms, you couldn't do that because everything is in one jar. But there's a method called single-cell RNA sequencing that Jan used to study 6 angiomyolipomas and 5 specimens of LAM. And other specimens of LAM were in parallel being analyzed at the University of Cincinnati. So this is one example of something that Jan found that we think is very important. So these are the cells in LAM, all separated now by color, depending on what kind of cells they are. So you can see here some epithelial cells, some cells that line the blood vessels, and then this big group of cells here, which are called macrophages. Now, macrophages are pretty interesting and important cells. Macrophage means to eat, a big eater. This is a depiction of a macrophage eating another cell. And I kind of think they're like the Roomba of the immune system. So they are traveling around looking for things that don't belong. In this case, looking for some dog fur on the carpet, but simultaneously not hurting things that belong there. So they're not going to try to eat the chair or hurt the dog. They're just looking for things that don't belong. The term macro and phage comes from the Greek for large and to eat. So they have a lot of jobs in the body. They are looking around for bacteria or tumor cells or debris of any kind. So there are actually two kinds of macrophages. There's one kind that I just told you about that are poised to get rid of stuff that doesn't belong there. But then there's a second kind of macrophages, the Roombas. There's a second kind of macrophage that seems to be turned off, basically. And these turned off macrophages are often found in tumors. We sometimes refer to them as tumor-associated macrophages, and they no longer are positioned to recognize and destroy tumor cells. So it turns out that in LAM, there are a lot of this kind of macrophage, which is unable to recognize and destroy a LAM cell. So in the future, one direction of this research could be that we can remove these B7H3 molecules from the surface of LAM cells, and that would allow the T cells to be active again and recognize and destroy the LAM cells. Another very exciting direction is that we could convert these tumor-associated macrophages, which are basically turned off, into the pro-inflammatory or Roomba-like macrophages that would then travel around the body looking for LAM cells and destroy them when they find them. So if we come back to the original question, can immune therapy eliminate LAM cells? We don't know. A lot more research is needed. But I think many of us in the field right now feel pretty excited that this could be a direction of the future, using our body's own immune cells to do what they are programmed to do, to recognize LAM cells as something that doesn't belong there and remove them. So we don't know the answer to this question, but what we do know for sure is that LAM research leads to breakthroughs. And let me just tell you a little more about these two amazing scientists who did the work that I showed you today. Tina Liu received a LAM Foundation grant for some of the work that she has pioneered on T cells. And she's gone on to establish her own research laboratory, which will include LAM research at Zhejiang University in China. And Yan Tang also received funding from the LAM Foundation for her work on macrophages and single-cell RNA sequencing that I showed you. And she was just promoted a few months ago to assistant professor of medicine at Harvard Medical School, and she will be continuing LAM research. So I feel very, very happy about the future of LAM research because the foundation has been able to get amazing scientists like this on a wonderful path toward future breakthroughs. Thank you very much. I'm going to stop sharing and turn it over to my wonderful friend and colleague, Steve Hummus.
Great, thank you. That was terrific. Thank you, Lisa. I mean, that was a great lesson in T-cell biology. I may ask you for your slides. So, I'm going to talk about something a little bit different, and but something that's also been on a lot of people's minds, and that's biomarkers and LAM research, and I call it Biomarkers Recent Advances. So, you know, we talk about developing medications or methods to try and slow down LAM or to really reverse it perhaps. But the problem is it's not so easy to track whether we're, how we're doing. And LAM is particularly tricky, right? Because if you have a solid tumor that you can follow by a CT scan or by a PET CT, you can do a treatment, you can see that the tumor gets smaller, you can say, okay, we're having success. But when you have microscopic adenomas or tumors that Lisa was talking about before, then you can't really track them by imaging. We have a cystic lung disease, but it's hard to really necessarily see that it's getting worse when it's already bad to start. And we can use FEV1 and pulmonary function tests to try and follow disease progression, but they're really functional assays and they're not getting at the root cause of LAM, which is these small tumors that are growing within the lung. So we need to find ways to follow tumor progression. And that's really what a biomarker is trying to find a biochemical way to track a disease, to A, perhaps use it to diagnose the disease, but B, also use it to track progression and also to track how well treatment is working. And so that's really a very important process as we're developing treatments for any disease, and in particular for LAM. We need really good, reliable biomarkers that work in as many patients as possible. So that's really, when we talk about biomarkers, what we're talking about. So let's start with the OG, the original biomarker, which was PMEL. You can notice I stole this from one of Lisa's papers, actually, a JCI paper from 2012. So there are a number of different cystic lung diseases, and when you're doing a CT scan, it's not so easy to tell them apart. Not every patient has genetic tuberous sclerosis, so sometimes it's difficult to know what this cystic lung disease is. And what was discovered many years ago was that the LAM cells express these melanocytic markers, these proteins that normally are found in melanocytes, but for some reason, they're showing up in these particular tumors. And we can measure them and use them to identify tissue that's consistent with LAM. So here's an example of a LAM lesion, and you can see that it stains, these little, the smooth muscle cell tumors are staining brown for PMEL. The antibody we use is HMB45. So this was a really useful way to diagnose LAM in a patient who had a cystic lung disease, and we weren't really sure what the cause was. But it's not a biomarker, right? Because you have to do a dangerous procedure in order to do this. You have to biopsy a cystic lung. You're not going to do it more than once. So you might do it at the beginning, but it's certainly not a method that we can use to track disease as it progresses. So it was a really helpful way when we were first learning how to make this diagnosis and starting to discover unusual proteins that seem to be upregulated specifically in LAM cells. So really the new kid in town, or the first and still the only really serum biomarker that's reliable that we can use for LAM, was discovered in the early 2000s, and that's VEGF-D. And this is the oldest paper I could find, which that really looked at this, and it was from 2006. And they found this protein VEGF-D. So what is VEGF-D? There's a bunch of different VEGFs, and they're basically proteins that normally their job is to help make and basically to help develop blood vessels and also to develop lymphatics. And so normally that's what they do, but it turns out a lot of tumor cells are really good at hijacking the human cells that are for normal physiology and using them for nefarious means. And in this case, they use them. A lot of tumors overexpress VEGF-D, which will increase blood vessels to the tumors, but will also increase lymphatics to the tumors. So really, it's a way to sort of create a highway that they can then go down to leave their original source and go elsewhere within the body. And that's really what VEGF-D does from a biological standpoint. But there are a lot of tumors that secrete VEGF-D, so it is indeed a biomarker, because the more tumor cells you have in general, the more VEGF-D production you're going to see. So in this initial study, you can see LAM patients on the left, and you can see control patients on the right. And on average, the LAM patients seem to have more VEGF-D than the control patients. So that's pretty exciting. Maybe we can just do a blood test and tell the difference between LAM and other cystic lung diseases. And I would say yes, if the levels are really, really high and there's no other tumors around, that's pretty good evidence that we're dealing with LAM. But you notice there's a lot of overlap. So it might be a good test for some patients who are up here, But as a diagnostic test, it's not going to be great because it's probably going to miss a third of the patients who happen to have lowish VEGF-Ds, right? So it's a biomarker. It's not necessarily a diagnostic biomarker, but as we'll talk about in a second, that doesn't mean that you can't use this biomarker to follow treatment in disease progression. So a biomarker can be diagnostic, but it can also be useful for following progression. All right, so we fast forward to a few more studies. This was one from Lisa Young and Frank McCormick we heard about before. And here's another study from Joel Moss's lab. Again, comparing healthy controls to LAM. You see pretty much the same thing. For some patients, pretty good, it's pretty high. But for a lot of patients, it's very close to what we see in healthy controls. So maybe see the same thing over here. So maybe not the perfect diagnostic test. But then the question is, could we use it to follow treatment? If it's working, when we treat with, say, sirolimus, we would expect to see the levels of VEGF-D go down in the serum of patients. And in fact, that's exactly what we saw in the MILES trial that Lisa just talked about. This is just comparing placebo to sirolimus. And what you find is that patients treated with sirolimus had a little over a 50% reduction on average in VEGF-D compared to the placebo treated. So it really does suggest that while VEGF-D may not be a diagnostic marker in all patients, it's a pretty good marker to follow progression. It seems to follow how well sirolimus is working, because we also know in these patients that you saw a really slow progression of disease. So whereas you saw that the progression of disease slowed in the MILES trial, here what you see is a 50% drop in VEGF-D. So what that tells us is that whatever treatment we were using, in this case sirolimus, is probably slowing down growth of these tumors quite considerably. And so the fact that FEV1 and other functions, lung functions, are holding steady, that's a good sign. It means the biomarker shows that the tumor cells are really slowing down and that we're holding disease progression to almost nothing. So this is a good example. VEGF-D, it turns out, is a pretty good biomarker. But the problem is that there are a lot of patients where it doesn't start out very high, and so you're not going to see a big drop. And there are some patients who you have very little VEGF-D at all. So it's just not going to be useful. So it's helpful. And then there are some patients who don't seem to have a drop in VEGF-D when you treat with a medication. So it's helpful, but we need more. We need other biomarkers that we can use together with VEGF-D to try and get a better feel for how well we're doing when we're treating a disease, rather than just do radio imaging and things like that. So what I'm going to do, this just shows you the sister of VEGF-D, which is called VEGF-C. This was also the MILES patients. This just shows you every single patient who is treated or placebo. And you can see in general the exact same pattern. In general, it looks like VEGF-C goes down. And then remember, after 12 months in the MILES trial, they stopped the treatment. And you can see that VEGF-C slowly starts to creep back up, which again is exactly what you'd want in a good biomarker. It goes down while you're treating, and it goes back up when you stop treating. So VEGF-D, VEGF-C, they're close sisters, and they serve similar functions in normal physiologic states, and they do seem to be secreted by tumors, and both could serve as potential markers or biomarkers. So again, we need to find some other biomarkers that we can use along with this to try and really cover all of our patients who have LAM. So I'm going to tell you a really quick summary of my story, which is very similar to Vera's story. So actually, I went to my first LAM Posium, I believe was 2011 or 2012. And that's the first time I met Vera. And actually, Lisa, you can either thank or not thank Lisa for inviting me to this meeting, because she is who got me interested in LAM. Never even heard of the disease before I really came to the meeting and learned from the physicians, the scientists, and the patients themselves. And I'm a reproductive endocrinologist. And so we were sitting around talking about the origin of the LAM cell. You know, you have these smooth muscle cell tumors. They seem to be estrogen sensitive. Essentially only women get LAM. And these tumors appear to be metastatic. And so the question was, well, what smooth muscle cell tissue is present in women, responds to estrogen, and could be metastatic? And the answer is women have a uterus, and a uterus has a myometrial layer of smooth muscle cells. And so the question was, could LAM cells actually be coming from the uterus? And so, like Vera, I applied for pilot funding and I think I got 50,000, Vera, just saying. But it was a few years later. It was a few years later. But that's what you need to make these mouse models. And so what we did is we knocked out TSC2 in the uterus. We eliminated expression of TSC2 in the uterus. And then we said, do the uterus develop LAM lesions or tumors? And do these tumors metastasize to the lungs? And the answer is they do develop lesions, LAM lesions, that have many, if not most of the features of LAM. And they do metastasize to the lungs. And in fact, all of this is very estradiol dependent in this model. So this is actually from the cover of Molecular Endocrinology. And this was work that was funded by the LAM Foundation. Since that time, we've continued to work on LAM. And I've received, like Vera, DOD and NIH funding. And if you look at that multiplication table, it's quite high. And it's really all because Lisa invited me to come to a LAM meeting. And we had this conversation about the origin of LAM. So that was work done by a really talented graduate student in my lab, Henske. And we published that in 2013. And now let's fast forward to 7 years later. You heard Lisa talk about single cell RNA sequencing. Well, the group from Cincinnati identified the LAM cells, the TSC2 null cells within their lung samples that they were examining. And they said, what do they look like? If we look at all the RNAs that are expressed, and we say, and then we compare them to RNA profiles of other tissues in the body, what do they look like? And in fact, what they found was by far and away, these LAM cells look like uterine cells. Number two, by a distant second, are smooth muscle cells. So 7 years later, we actually saw some evidence to suggest that indeed these cells in the lungs could be coming from the uterus. Now I say could be. It's also possible that they're not coming from the uterus, but they just look like uterine myometrial cells. And quite frankly, either answer is interesting, and either answer is consistent with what we were finding in our mouse model, and kind of gave us confidence that the mouse model was somewhat useful. And it's a great example of what Vera mentioned before about how these preclinical mouse models that we do, sometimes a little bit rolling the dice, can actually lead to some really interesting results that apply to human disease. And when we talk about GPNB, it's going to be another example of that. So I'll go through it pretty quickly. I just want to show you, this is what a normal uterus is. A little different than a human uterus. You have these two horns. And at 18 weeks, this is a knockout uterus. You can tell the difference in size. And the knockout uterus has all these white things are all myometrial tumors. They all express melanocytic markers. They're TSC2 null. They have estrogen receptors, progesterone receptors, a lot of the features that we see in LAM. If you eliminate the ovaries, you do an oophorectomy and remove the ovaries at 18 weeks. By 28 weeks, look what happens to the tumors. They pretty much go away. We see the same thing if you treat with a drug called letrozole, which inhibits the enzyme that makes estrogen. So we get rid of the estradiol by preventing it from being made by the ovary, and we see pretty much the same cell. Same result. These are not normal uteri. They do have these cysts in them. They're still not normal, but they certainly don't look like this, right? So it really does show you the incredible estrogen sensitivity that we are seeing in our in these mice. So armed with this, we did a different study than single cell RNA sequencing. We were a lot simpler, and back then single cell RNA sequencing was not so easy to do. We just did bulk RNA sequencing, comparing this uterus to this uterus. And then we can also compare this uterus plus estrogen versus this uterus plus estrogen. And we were looking for genes that are upregulated in the absence of TSC2, so when mTOR is turned on, but even more upregulated when you added estradiol, so the super upregulated genes. And top of the list were all the melanocytic markers that I mentioned at the beginning. PMEL is right here. And then we have 4 others here. There are several others that we found as well. And so the squares are the knockouts and the circles are the wild type. So most of these are really not expressed in a normal uterus at all. And as the mice get older, they get more and more expressed in the uterus. So again, here's PMEL, the one that I showed you that we used to use to diagnose by biopsy. But the one that really interested us was this one called GPNB, because it's really not in the normal uterus at all. It goes way up as the mice get older. And let me tell you, it's the most estrogen sensitive one. It really goes up in the presence of estradiol. So what is GPNB? It's normally expressed in the cytoplasm of melanocytes. You also find it in those cells that Lisa mentioned called macrophages. But when a melanocyte, benign melanocyte, becomes a melanoma, it goes from the inside of the cell to the outside of the cell. It gets expressed on the cell membrane. So only when you have a cancer is GPNMB on the cell surface. Otherwise, it's inside. And what's interesting is when it gets to the cell surface, it actually releases an ectodomain. It releases about 2/3 of its protein are actually released from the cell and go into the media, for example, when you have a tissue culture dish with these types of cells. So it gets released. And in fact, we identified where it's getting cut, and we also identified what's cutting it. So we know how it's getting cut and where it's getting cut. And GPNMB is associated with aggressiveness in a number of tumors. And again, since it's only on cancers, it's only on the cell surface in cancers, it's an interesting and potential drug target. This just shows you some TSC2 null cell lines that we commonly use in our labs. And I just want to point out that there's intracellular GPNMB, but here it is at the membranes. The arrows are pointing to it. You can see it there. And this cell line, this is a human cell line, which we got from Lisa, actually. You can see him here. Okay, so it's right at the cell surface, just like in melanoma. Okay, this just shows you staining by immunofluorescence our knockout uterus versus a normal uterus. There is no GPNMB in the normal uterus, a lot of GPNMB in the knockout. If we get rid of the ovary, you can see a few specks of green, but there's virtually no GPNMB. So it's very estrogen sensitive, and you only see it in the knockouts. So we found this in a mouse. So then we wanted to turn to human tissue. And this just shows you a human lung, and this is a LAM lesion. And you can see that it lights up beautifully for GPNMB. This is actually a uterine LAM lesion. This is inside a uterus. Here's the lesion. Here's normal uterus. And again, you can see that the lesion lights up beautifully. LAM lights up beautifully, but not the normal uterus. And so this is at least as good, if not better, than PMEL. It's a really bright signal. It's easy to see. But again, it doesn't help us because it's not a real biomarker because we have to get tissue in order to do this. But remember, I mentioned that when it's on the cell surface, it gets released from the cell surface. So we wondered, well, if it's getting released from the cell surface, maybe we can find it in the blood of patients with LAM. So can GPNMB be used as a biomarker for LAM? And this is work that was just published by a really talented graduate student, Erin Gibbons, standing in front of her poster here at a meeting. I think that was actually the LAM meeting when she presented this. And here's what she found. LAM, these are actually samples that we got just to show the collaborative nature of what goes on in LAM. These are samples that we actually got from Lisa. And they're LAM patients versus controls. And you can see that just like VEGF-D, that on average, there's more GPNMB in the serum of LAM patients versus controls, but just like VEGF-D, there's an overlap. So if it's high, it's pretty useful, but if it's normal, you can't use it as a diagnostic marker, but could you use it as a way to track therapy? So Lisa gave us about 10 patient samples before and after sirolimus treatment, and you can see that in fact, in almost every patient, except for really 2, we saw a reduction in GPNMB when you treat with sirolimus. So then we went back to Frank McCormick, and he was generous enough to actually look at all those LAMs, all those MILES trial patients to follow GPNMB, just like I showed you earlier for VEGF-D. And this is just the average values. So here's the placebo, and here's the sirolimus treated. And you can see that VEGF-D GPNMB very significantly decreases when you treat with sirolimus. When you stop the treatment at the 12-month point, it starts to creep back up again. But even at 6 months, there's dramatic separation. So we think that this is exactly, again, what you want to see in a biomarker. It's pretty significantly reduced. It seems to be happening in almost all the patients that are treated with sirolimus. And it really does correlate with treatment. When you stop the treatment, it starts to creep back up. So it's not the be-all, end-all. Because like VEGF-D, there's patients where it's not going to work. But we're starting to get somewhere when you have 2 biomarkers. And maybe we want to have 3 or 4 biomarkers. And we can formulate an algorithm where we can use them all together as a really significant and useful way to track disease progression over time. So it's exciting that we're moving in the right direction, but obviously a lot more has to be done. And we need to find more and more specific biomarkers, again, so we can utilize them all together to try and really be able to follow disease progression in every patient who has LAM. For some patients, one biomarker may turn out to be better than another, but we really need to have several options that we can choose from. So, conclusions, it's a potential marker for the diagnosis and treatment success of LAM. GPNB is a potential drug target. I didn't really talk about its functional components, but it turns out it really is important for tumor growth. Again, may not be the be-all end-all, but it's proof that preclinical mouse models actually can have a great value. And we really need to have basic science research collaborating with clinical research if we're really going to make progress here. And again, to go back to what's been said from Vera and also from Cindy, this is another example supporting how LAM Foundation funds and research money can go towards programs that can make a difference. I've been in this now since 2013, and I can't imagine doing anything else with my life. And almost all of my lab now is studying LAM. And it's really because of people like Vera and people like Cindy and people like Lisa and Frank McCormick, who wrote me in and then showed me how important it is to work at every level with the LAM Foundation. So I'm really appreciative that all of you are here. And I will stop sharing my screen and we can do some questions.
Wow, thank you both for this enlightening discussion this evening. I was especially struck by just like Lisa illustrated, Dr. Henske, sorry, illustrated the out-of-the-box thinking when it comes to biomarkers, when it comes to finding new ways to destroy those LAM cells. And as somebody who was here back in the day, when we were simply talking about sirolimus, the expansion of our thinking and progress is really inspiring. So thank you both. At this point in time, we will go ahead and open the chat box for questions. And I'm going to go ahead and begin with a few questions submitted to us in advance. So one of the kind of broad questions that our patients have is how are these topics for research chosen? What motivates you? What drives you? What is kind of the overarching impetus for the way you approach your research?
Maybe I could just start by saying
Sure.
that our work in immunology, the immune cells and the T cells, was sparked by a question from a woman with LAM who was wondering about whether these treatments that work so well for cancer might also have some role in LAM. So often the questions that we are addressing scientifically are the ones that come to mind if you're someone living with LAM.
And I would echo that. As I mentioned, really the start of my research in LAM was really just coming to a LAMposium and just being engulfed in it all and having that conversation about the origin of the LAM cell. But the other thing I'll say is, there's that expression, I think somebody said that chance favors the prepared mind. And I would say that in most of my research, if you were to look at my CV, it's not one straight line at all. And I've really gone, and so I do a lot of reproductive endocrinology, but the reason why is because when I started out, I was studying steroid signaling and a frog oocyte. And it happened to be that oocytes obviously are involved in reproduction. And the way the science went took me into reproduction when that wasn't where I started. So a lot of where I've ended up in everything, including LAM, you start with an idea from a patient or from an organization, and then something just pops up and you just want to look at it further. And that leads to another discovery.
Right. I love that. We have a question in our chat box that says, are there any plans to study CRISPR and LAM or gene editing?
Not to my knowledge, but the oncologist could probably.
I think it's a very interesting idea and really looking forward to how we could be modifying the gene defect. We know the gene defect in LAM, but I don't think we're there yet.
It's a difficult disease because, as you know, in tuberous sclerosis, every cell has only one deletion when you start, and then you have this loss of the second allele, and it's impossible to predict where that's going to happen or when it's going to happen. So it would be difficult. It'd be easier in a disease that's in the bone marrow where you can just take out the old and put in the new. But what I will say is, and I'm sure Lisa and Vera do the same, we use CRISPR-Cas in our lab all the time to knock out genes in cell lines, to knock out genes in mice. So even if we're not using it in patients, it's a technology that's revolutionized the way we do science.
Great. I'm wondering, particularly Dr. Henske, with the information that you presented this evening about the macrophages and the T cells, one of the questions that was asked was, given what scientists know now about LAM, its diagnosis, treatment, and progression, what seems most promising in looking forward to an eventual cure? You mentioned these specific things, but are there ways that we can actually do that, right? Internally, so.
Right. Yeah, I'm always nervous about over promising because we definitely see directions that we could use to, for example, eliminate that B7H3 protein from LAM cells, but any treatment takes quite a long path to develop. We were so lucky with sirolimus because it was already FDA approved and actually already had a safety track record, even in children. And that made it faster for us to move that forward in the MILES trial. Many of the other treatments are under development, but are still years away.
Right. Okay, thank you. I have a question from the chat box. Is it possible to get T cells that are not exhausted from donors, like a blood donation or a bone marrow transplant?
Well, I think that's an interesting idea, and theoretically it could work, but there are much easier ways to do it, which would be to, for example, use one of the treatments that are being used in cancer, or for example, to sort of turn down or turn off this B7H3 protein. Then it's your very own T cells that are now able to recognize and destroy the tumor cells, not someone else's where there could be autoimmune reactions and, what we call graft versus host disease. So yes, that could work. And it's good to have these ideas coming forward.
Right.
We think we can do it more safely than that.
Yeah, as Lisa mentioned, there are already other ways that T cells get exhausted, and there are antibodies we can treat patients with for cancers that for some work better than others to try and turn them back on. But I do think it's I think it's a really important and interesting. I think it is the future for a lot of tumor biologies, to try and get our own body to destroy the cells, because it seems like a logical and safe way to do things if you can do it properly.
Sure, absolutely. And then another question, and this is really along the estrogen line of questioning. Have studies been done on confirmed LAM patients who are either post-menopause or who perhaps were receptor-positive cancer patients and had both their uterus and ovaries removed, followed by aromatase inhibitor therapy?
That is a really good question. I could talk about it forever, but I'll try to be short. So there's never been a real clinical trial to look at to see whether eliminating estrogen production or actions has an effect in women with LAM. There was a trial that we did that I was part of, but it was really Frank McCormick's group and others that took a look at this, but they did it in postmenopausal women. And guess what? There was no difference because the disease didn't really progress very much in any of the women, probably because it was postmenopausal and estradiol levels were already low to start. So it is an interesting question. It's a clinical trial that has yet to be done, and it's something we talk about a lot, but we still haven't done it. It's certainly something at some point that I think needs to be tested. There's a lot of anecdotal descriptions of people where they're treated with aromatase inhibitors or their ovaries are removed and they see improvement in LAM. And there are some LAM patients who their LAM seems to get worse in cycles that seem to go along with the menstrual cycle. And there are people in Cincinnati who are studying that as well. And maybe those will be the really estrogen sensitive group that you'd want to do this clinical trial on. But it's a question that, interestingly, after all these years, has not really been studied in humans yet.
Right. And then to dovetail on that, we hear a lot about the role of estrogen in LAM. Are there any plans to understand more of progesterone's role?
So progesterone is tricky because it has a lot of other effects in the body, and so sometimes people feel badly when their progesterone levels are high. Everybody's progesterone is high just prior to menstruation when you're cycling, and it's actually what is considered to be the steroid that makes you feel bloated and miserable and tired and hard, gives you your brain fog. So it's a difficult hormone. So most of the evidence suggests, though, that it isn't progesterone because, again, it's anecdotal, but there have been a lot of individuals who are put on progesterone, depot progesterone, to try and actually lower ovarian production of estrogen. And again, anecdotally, many of these patients get better. So, and the thought is, therefore, it's probably more likely the estrogen than the progesterone, also because we know estrogen drives the growth of other tumors, whereas progesterone hasn't really been shown to drive the growth. But we don't know for sure. And again, there's always exceptions. And so maybe for some individuals, progesterone is a player in this, but it's always tricky with progesterone because it does so many other things that it's hard to separate it out.
Right. The more we learn, the more we need to learn, right?
Yep.
We just keep growing in research and knowledge. So, and I'll end with this question. This speaks again to the T cells. Can you program alert T cells with your own stem cells for stem cell injection therapy?
So could we, I guess, give back someone's own cells and allow them to reprogram? Yes, you probably could, but again, that might be more, have more side effects and more intervention than allowing the T cells to readapt themselves. So we know that T cells can go from, and macrophages can go between different cell states, and they don't have to be sort of reborn to do that. We think they can remodel themselves, kind of. So yes, that would be another idea that we could consider.
Great. And then finally, we are a very active community, whether we're LAM patients or scientists or clinicians. And so my final question would be, what specific actions can patients do to help further LAM research?
So I'll start, and then Lisa can get the last word. I think we're both going to say the same thing, though, and that is to stay involved, to participate, to let us know what you're thinking and what you want us, what's most important to you. But also when there's opportunities to be involved in clinical trials or basic science where they need blood or whatever, to volunteer if you can. And I think that's what amazes me about the LAM Foundation and the women who are involved in the LAM Foundation, how willing they are to participate. I can't think of another part in my medical or scientific life where I see anything that comes close to this.
Dr. Henske, any final words?
Couldn't say it better. I think we have a wonderful community, and I guess one thing that you can do is to help raise visibility of LAM research, because we think there are many women who don't know they have LAM, and many providers who are not that familiar with LAM. So every time you talk about LAM to a friend or a colleague or at an event, you're helping the world understand more about LAM, and that will help the research, help get more people involved.
Wonderful. Thank you. And somebody just typed into, Lisa Pascal, our Circle of Hope coordinator, said, and tissue donation. We all need to really be thinking about as patients tissue donations. So thank you again, Dr. Krymskaya, Dr. Henske, and Dr. Hamas for helping us understand the progress being made in LAM research and how we can contribute to it. You all inspire us. You continue to motivate us to continue to get the word out and to raise those funds. So thank you for being here. I know your time is busy, and we so appreciate you.